There is a word you have forgotten. Not a word you never knew. A word that lived in your mouth, ordinary and reliable, until one afternoon it simply vanished. You reach for it in conversation, and there is only silence. A moment passes. The word returns, but not before you have felt something shift. Something small, but undeniable. This happens more often now. Not just words. Names of people you have known for years. The reason you walked into a room. The thread of what you were saying 3 sentences ago. You smile and move forward, attributing it to stress, to age, to the simple accumulation of living. Your doctor checks your blood work. Everything is fine, they say. Your cholesterol is acceptable. Your glucose is normal. You are fine, but you do not feel fine. You feel as though you are thinking through water. By mid-afternoon, the fog thickens. There is a heaviness behind your eyes, a fatigue that coffee cannot penetrate. Your mind becomes sluggish, your focus scattered. You push through it, as you always do, but you notice it. And in noticing, you begin to wonder, is this what aging actually feels like? Is this inevitable? Is this simply what happens when you pass 40? Millions of adults are asking this same question in private. They do not mention it to their doctors because they have learned that doctors will find nothing measurably wrong. They do not discuss it with friends because it feels like a personal failing, a sign that their mind is not what it once was. So they carry this fog silently, adapting their lives around it, scheduling important decisions in the morning, avoiding complex conversations in the afternoon, accepting that mental clarity is now a luxury of youth. What if I told you that this fog has a name? What if I told you that it is not inevitable, not irreversible, and not actually about aging at all? What you are experiencing is real. The cognitive decline is measurable. The afternoon collapse is genuine. But the story you have been told about why this is happening, that your brain is simply aging, that this is the price of time, that story is incomplete. There is another possibility, one that changes everything about how you understand what is happening inside your skull. Your brain consumes roughly 1/5 of all the energy your body produces at rest. It is the most metabolically demanding organ you have, and like every organ, it requires fuel. That fuel is glucose. But here is where the story becomes crucial. The way your brain accesses glucose has changed. The doorway that opened effortlessly in your 30s has begun to close, and the fog you feel is not your brain aging. It is your brain starving. This is not poetic language. This is metabolic reality. And to understand it, we must trace a story that begins not in your neurons, but in your liver. In the way insulin, the hormone that should be your brain's greatest ally, has gradually become its obstacle. You have been told that insulin resistance is a disease of the overweight, a precursor to diabetes, something that happens to other people. But insulin resistance is far more common than that narrative suggests, and its effects on the brain are far more immediate than most people realize. It can be present in people of normal weight. It can exist without elevated blood glucose, and it can be silently reshaping your cognitive function for years before you even know it has a name. The word that dissolved mid-sentence did not disappear because your neurons are dying. It disappeared because those neurons could not access the fuel they needed to fire properly. The afternoon fog is not fatigue, it is energy crisis. And the fact that your doctor said your numbers are fine is not reassuring. It is actually the point. This is happening beneath the threshold of standard medical testing. This is the threshold moment, the place where understanding mechanism becomes the pathway to reversal. Everything you are about to learn, about glucose transporters, about insulin signaling, about neuroinflammation and tau phosphorylation, is not abstract neuroscience. It is the explanation for why you feel the way you feel. And more importantly, it is the map showing how to feel differently. Before we continue deeper into this investigation, subscribe to the channel and tell us in the comments where you are watching from. Because what comes next will change how you understand not just your aging, but your agency in it. Now you understand why this matters. Your brain does not simply age like the rest of your body. It ages according to the fuel supply it receives. And that fuel supply is directly controlled by a single hormone, insulin. In your 20s and 30s, the system worked with remarkable elegance. When you ate a meal containing carbohydrates, your blood glucose rose. Your pancreas sensed this rise and released insulin proportionally. That insulin circulated through your bloodstream, arriving at cells throughout your body with a simple message: open your doors and take in glucose. Your brain cells listened. Glucose entered. Energy was abundant. Neurons fired with precision. Memory consolidated. Your mind felt sharp. This was not a process you were aware of. It happened silently, billions of times per day, like a water system that works so well you never think about the pipes. But those pipes, the biological machinery that allows glucose to enter your brain cells, are specific and finite. Understanding them is the key to understanding what changed. Your brain relies on specialized proteins called glucose transporters. Think of them as molecular gates embedded in cell membranes. GLUT1 is the constitutive transporter, meaning it works all the time, always allowing some baseline glucose entry. But GLUT4 is different. GLUT4 is insulin dependent. It sits dormant inside the cell until insulin arrives and signals its arrival. When that signal is received, GLUT4 proteins relocate to the cell membrane, creating new doorways for glucose to enter. This system is elegant precisely because it is responsive. More glucose in the blood means more insulin, which means more GLUT4 transporters at the membrane, which means more glucose gets into the neurons that need it. Supply meets demand perfectly. This responsiveness is what allowed you to think clearly through a challenging workday. This is what enabled you to study for hours, to solve complex problems, to hold multiple threads of thought simultaneously. Your brain had access to fuel on demand. The system was efficient. The communication between your pancreas, your bloodstream, and your neurons was seamless. You took this for granted because you had no reason not to. But here is what happens over the course of years, usually without your awareness. The system begins to respond less to insulin's signal. It is not that insulin disappears. It is that the cells receiving insulin's message become less responsive to it. Your pancreas, sensing that glucose is not being taken up as effectively, responds by releasing more insulin. More insulin means higher insulin levels in your blood. But if cells are not listening to the signal, more of the signal does not solve the problem. It only amplifies the stress on the system. This process affects nearly every tissue in your body, but the brain experiences it in a particular way. Unlike muscle or liver tissue, which can use alternative fuels more readily, your brain in your earlier decades has become dependent on glucose. It has specialized in running on this fuel. When insulin resistance develops, the neurons cannot access glucose as effectively. But the glucose is there in your bloodstream. It cannot enter, it is locked outside. The paradox is crucial. You can have elevated blood glucose and starving neurons simultaneously. You can have high circulating insulin, your pancreas working overtime, and your neurons still not receiving sufficient fuel. This is not a failure of the glucose. This is not a failure of the insulin. This is a failure of communication. The signal is being sent. The receptor is not receiving it. And here is what your brain does when it cannot access adequate glucose. It activates a cascade of responses designed to signal distress. It calls for help. The cells that oversee brain immunity respond. Inflammation increases. Proteins begin to misfold. Energy production in the mitochondria becomes inefficient. Your neurons, in essence, begin to suffocate despite living in a sea of fuel they cannot access. This is the metabolic foundation of the fog you feel. This is why your mind feels sluggish in the afternoon. This is why the word dissolves mid-sentence. Your brain is not aging. It is being starved of the fuel it has learned to depend on. And the tragedy is this. At your age, with your metabolism, this is almost certainly reversible, but only if you understand it. Only if you see the mechanism clearly enough to interrupt it. Everything that follows, the neuroinflammation, the tau hyperphosphorylation, the synaptic decline, the accelerated cognitive aging, all of it flows from this single rupture in the relationship between insulin and the glucose transporter. This is where we must look next. When insulin stops listening, the fundamental dialogue between your hormone and your cells becomes garbled. This is not a sudden event. It is a slow erosion of responsiveness, a gradual dimming of the signal that has orchestrated your metabolic harmony for decades. To understand what happens next, you must first see what is happening at the cellular level, where insulin resistance is being written into your biology. At the heart of insulin resistance lies a disruption in what scientists call the insulin signaling cascade. When insulin arrives at a cell and binds to the insulin receptor on the cell's membrane, it should trigger a precise chain reaction. That receptor activates a protein called IRS1, insulin receptor substrate 1. IRS1 is like a relay runner receiving the baton of the insulin message. It should pass that signal to the next runner in the chain, a protein complex called PI3K, which then activates ACT, a kinase that initiates the glucose uptake machinery. This entire sequence should unfold in milliseconds. It is elegant molecular choreography. But in insulin resistance, something interrupts this dance. The insulin receptor becomes less responsive. More commonly, IRS1 itself becomes serine phosphorylated, marked by a chemical modification that blocks the signal cascade. It is as though the relay runner suddenly cannot grip the baton. The message is still being sent, but the pathway is jammed. The PI3K ACT signaling cascade becomes attenuated. The signal weakens. The glucose transporters do not relocate, and the cell, still needing energy, does not receive it. This mechanism is well understood in muscle and liver tissue, where it contributes to elevated blood glucose and the metabolic dysfunction associated with type 2 diabetes. But the brain is different. The brain is not only different, it is particularly vulnerable to this disruption because of how it has evolved to feed itself. Your brain expresses insulin receptors on its neuronal membranes. It participates in the insulin signaling system. And because neurons, unlike muscle cells, have limited capacity to use alternative fuels efficiently, in the short term, they depend on consistent glucose availability. When systemic insulin resistance develops, when your liver cells and your muscle cells stop responding to insulin, that resistance is not confined to those tissues. It spreads systemically. It is a whole-body phenomenon. And the brain, despite being protected by the blood-brain barrier, cannot escape it. Here is what makes this particularly insidious. Your pancreas does not know that your cells have become resistant to insulin. What it knows is that glucose is not being taken up as efficiently as it should be. The pancreas interprets this as insufficient insulin and produces more. You end up in a state of hyperinsulinemia, abnormally high circulating insulin, combined with insulin resistance. Your bloodstream is flooded with a hormone that your cells are increasingly ignoring. The signal is louder, but the receiver is not listening any better. For your neurons, the consequence is severe. The glucose in your bloodstream cannot enter the cells that need it most. The GLUT4 transporters, which should be relocating to the cell membrane in response to insulin signaling, remain largely dormant. Glucose uptake into neurons declines, not because glucose is absent, not because the neurons are broken, but because the communication system that allows neurons to access glucose has become degraded. This state, where glucose cannot enter the brain efficiently despite being present in the blood, where circulating insulin is high but cells are not responding, is sometimes called cerebral insulin resistance. In its more severe forms, when it progresses to neurodegeneration, it has been termed type 3 diabetes, emphasizing that Alzheimer's disease may be fundamentally a disease of brain glucose metabolism gone wrong. But here is what must be emphasized. At this stage, before neurodegeneration has occurred, before plaques and tangles have accumulated to the point of irreversible damage, this is not a disease. It is a metabolic miscommunication. The system has not broken. It has become insensitive. And insensitivity can be restored. This is the critical distinction that changes everything. Your neurons have not forgotten how to take up glucose. The GLUT4 transporters have not disappeared. The signaling machinery is still there. What has happened is that the constant barrage of high glucose and high insulin has, over years, made your cells less responsive to the signal. The system has adapted to ignore an overwhelming stimulus, the way you stop noticing a sound you hear every day. But this adaptation is not permanent. It is not hardwired into your biology. It can be reversed. And the reversal begins when the signal, the glucose, the insulin, returns to the normal range. When that happens, cells begin to listen again. What occurs next, however, is where the story becomes darker. Because while your metabolic system has been struggling to respond to insulin's signal, something else has been happening inside your neurons. And that something, once it begins, creates a cascade of events that demands your attention. When neurons cannot feed, the energy crisis becomes real. Deep within each nerve cell, the mitochondria begin to fragment. These are the power plants of the cell, the structures that convert fuel into the chemical currency of life. ATP, adenosine triphosphate. Without adequate glucose, mitochondrial efficiency plummets. The membranes that once held tight, generating the electrical potential necessary for thought, begin to deteriorate. ATP production declines. The neuron grows darker, more desperate. This is not a gradual inconvenience. This is a biological emergency signal. And your brain has sensors for emergency. When neurons sense this energy depletion, they do not suffer in silence. They cry out. They release molecular distress signals, chemical messengers that alert the immune cells of the brain that something has gone wrong. Enter the microglia. These are the brain's immune sentinels, the cellular guardians that evolved to protect neural tissue from infection and clear away debris. They sit quiet and vigilant in the spaces between neurons, waiting for a signal that threat has arrived. When energy-stressed neurons release their distress signals, including molecules like ATP itself and danger-associated molecular patterns, the microglia sense it immediately. They activate. They transform from resting sentries into aggressive warriors. This activation, in small doses and for short periods, is protective. Microglia that activate and then return to rest perform essential housekeeping. They clear away damaged proteins and dead cells. But here is what happens when the stress is chronic, when it persists month after month and year after year. The microglia do not rest. They remain in a state of chronic activation. And chronically activated microglia do not clean. They inflame. They begin releasing cytokines. These are signaling molecules that coordinate immune responses, and they include IL-6, tumor necrosis factor alpha, and other inflammatory mediators. These cytokines are meant to be brief signals, temporary messengers that appear for minutes or hours to organize an immune response. But when chronically activated microglia release them continuously, these short-term signals become chronic stressors. They bathe the surrounding neurons in an inflammatory environment that was never meant to exist. The neurons, already stressed from energy depletion, now face an additional insult: neuroinflammation. They are surrounded by chemicals that damage them. Synaptic connections, the bridges through which neurons communicate, begin to weaken. The proteins that hold these connections together start to degrade. And it is here that something more sinister begins. It is here that tau enters the story. Tau is a protein found abundantly in neurons. Its normal function is structural. It stabilizes microtubules, which are like the skeleton of the cell, providing its shape and allowing transport of molecules within it. But tau is also exquisitely sensitive to phosphorylation, a chemical modification where phosphate groups are added to specific sites on the protein. Normally, tau is phosphorylated lightly and transiently. Phosphatases come along and remove these phosphate groups, maintaining balance. But in neurons that are chronically energy-stressed and chronically inflamed, this balance collapses. The kinases, enzymes that add phosphate groups, become overactive. The phosphatases cannot keep up. Tau becomes hyperphosphorylated. With too many phosphate groups attached, tau changes shape. It can no longer bind properly to microtubules. Instead, it begins to aggregate with other hyperphosphorylated tau molecules. The tangles begin to form. These neurofibrillary tangles, twisted formations of hyperphosphorylated tau, are hallmarks of cognitive decline. They accumulate in brain regions critical for memory and learning, particularly the hippocampus and entorhinal cortex. They represent neuronal dysfunction at the molecular level. But tau hyperphosphorylation does not work alone. The same chronic neuroinflammation that drives tau pathology also drives the accumulation of amyloid beta, another protein associated with cognitive decline. These two pathologies feed each other in a vicious cycle. Amyloid beta activates microglia further, which increases inflammatory signaling, which drives more tau phosphorylation, which itself triggers more microglial activation. And as this cascade accelerates, something else is lost. The neurons, battered by chronic inflammation, produce less BDNF, brain-derived neurotrophic factor. This is perhaps the most important protein for maintaining neuroplasticity, the brain's ability to form new connections and rewire itself. BDNF is like fertilizer for the brain. Without it, learning becomes harder. Memory consolidation weakens. The brain loses its adaptability. The cognitive decline that begins as afternoon fog and word-finding difficulty becomes over years of this metabolic siege, something more entrenched. But here is what must be understood. This entire cascade began with one reversible problem. It began with insulin resistance, and if that problem is addressed, the cascade can be interrupted. Part 5. But there is something else your brain possesses, an ancient metabolic flexibility that most adults have forgotten exists. Your brain can run on more than glucose. It has always had this capacity. Your neurons can feed themselves on ketones, molecular fragments that are produced when your body metabolizes fat in the absence of abundant carbohydrates. This is not a new discovery. This is not experimental medicine. This is basic biochemistry, known for over a century, yet largely forgotten in the age of constant carbohydrate availability. Ketones are elegant fuel. They move through your bloodstream and cross the blood-brain barrier with ease. Once inside neurons, they enter mitochondria directly and are converted into ATP with extraordinary efficiency. In fact, ketone metabolism produces more ATP per unit of oxygen consumed than glucose metabolism does. Your neurons, when running on ketones, are more powerfully energized than when running on glucose alone. But here is what makes ketones revolutionary in the context of insulin resistance. They do not require the broken system to work. They do not need GLUT4 transporters. They do not depend on insulin signaling. They bypass the entire mismatch between circulating glucose and neuronal uptake. When your brain runs on ketones, it is no longer starving while surrounded by plenty. It is fed directly and completely by an alternative fuel source that your body can generate whenever carbohydrate intake drops. The consequences are profound. Beyond merely providing energy, ketones have anti-inflammatory properties that act directly on the microglia. When ketones are available, microglial activation decreases. The cells that have been bathing your neurons in inflammatory cytokines begin to quiet. IL-6 and TNF-alpha production drops. The chronic inflammatory state that has been driving tau hyperphosphorylation begins, gradually, to reverse. There is more. Ketones suppress the production of amyloid beta. They enhance the expression of BDNF, that precious protein that fertilizes neuroplasticity and learning. Under ketone-dominant metabolism, neurons begin to repair connections that have been weakened by years of energy stress and inflammation. The brain's capacity to form new memories, to learn, to adapt, begins to return. This is where the hippocampus enters the story again. The hippocampus, that seahorse-shaped structure buried deep in your temporal lobe, the seat of memory formation, shows something remarkable under ketone-dominant metabolism. It generates new neurons. Adult neurogenesis, the birth of new nerve cells even in aging brains, is enhanced when ketones are the primary fuel. Your brain is not locked into stasis. It is not doomed to accumulate damage. Given the right metabolic conditions, it actively rebuilds itself. The synaptic plasticity that had been collapsing under chronic energy stress and inflammation begins to restore. Long-term potentiation, the neurochemical process by which memories are encoded into persistent changes in synaptic strength, becomes more robust. The brain's fundamental ability to change in response to experience returns. This is the bridge between understanding the problem and becoming the agent of your own recovery. Your brain has not lost its capacity to heal. It has lost access to the fuel that allows that healing to begin. That access is not gone. It can be restored through something far simpler than any pharmaceutical intervention, through the restoration of metabolic flexibility. Metabolic flexibility is the ability of your body and brain to switch between fuel sources, to run on glucose when carbohydrates are abundant, to run on fat and ketones when they are not. Insulin resistance destroys this flexibility. It locks your brain into glucose dependence while simultaneously making glucose inaccessible at the neuronal level. But metabolic flexibility is not a trait you either have or lack. It is a capacity that can be recovered. The path to that recovery begins with addressing the root cause, the insulin resistance that created this mismatch. When insulin resistance is reversed, metabolic flexibility returns. The brain's ability to access both glucose and ketones is restored. The ancient neural machinery that has existed since before humans invented agriculture awakens. What happens next? In the weeks and months that follow the restoration of metabolic flexibility, is a cascade of recovery as powerful as the cascade of decline that preceded it. The energy crisis resolves. The inflammatory state recedes. The brain begins to remember how to be itself again. And this is not theory. This is measurable, observable, repeatable. This is the demonstrated pathway through which cognitive decline becomes cognitive restoration. What reversal requires is not a dramatic intervention, but a sustained shift in what you consume. Refined carbohydrates and processed sugars, the dietary staples that elevate blood glucose and demand constant insulin response, must be reduced. This is not deprivation. This is precision. When carbohydrate intake normalizes, insulin levels begin to normalize with it. The hormone that has been circulating in excess, hammering on increasingly deaf cellular receptors, begins to retreat to physiological levels. Over weeks and months, something biochemical shifts. Cellular insulin sensitivity returns. Inside the hepatic cells and muscle tissue where the resistance first took root, IRS-1 serine phosphorylation decreases. The molecular gate that was blocking the signal begins to unlock. PI3K ACT signaling becomes responsive again. The cascade that had been interrupted starts to flow. This is not instantaneous. The body does not reverse years of metabolic dysfunction in days. But it does reverse. The capacity for restoration is built into your cellular machinery. And what happens in the neuron during this reversal is equally precise. As insulin signaling improves throughout your body, it improves in the brain as well. GLUT4 transporters, which had been stranded in intracellular compartments, unable to respond to insulin's summons, begin to relocate to neuronal cell membranes. The doors begin to open. Glucose uptake into neurons increases. The paradox resolves. No longer is your brain awash in circulating glucose while its cells starve. The energy crisis that had silently accumulated begins to lift. But the reversal does not stop at glucose uptake. As the energy stress on neurons decreases, something shifts in the immune dialogue happening inside your brain. Microglia, those sentinel cells that had been activated by years of neuronal distress, begin to quiet. The constant state of alert begins to ease. IL-6 and TNF-alpha production declines. The inflammatory cytokine bath that had been corroding your synapses begins to drain away. The chronic neuroinflammatory state, that silent fire that had been burning through your cognitive reserve, begins to cool. And as it cools, the tau pathology itself begins to reverse. Inside neurons, the serine and threonine kinases that had been hyperphosphorylating tau slow their work. Phosphatase activity, which removes those phosphate groups, becomes more robust. The tangled tau protein, accumulated over years, begins to dephosphorylate. The neurofibrillary pathology that had been accumulating does not disappear overnight, but it plateaus. The neuron begins to clear what had been poisoning it. The synaptic connections that had weakened under the weight of tau tangles remain compromised, but they are no longer being further damaged. The amyloid beta accumulation also slows and begins to reverse. As microglial activation decreases, the inflammatory process that drives amyloid production becomes less aggressive. The vicious cycle between tau and amyloid beta, where each pathology triggers more of the other, is interrupted. The brain's own clearing mechanisms, long suppressed by chronic inflammation, become active again. And perhaps most importantly, the production of BDNF increases. As the inflammatory environment around neurons improves, as energy availability stabilizes, neurons begin to produce more of this fertilizer for neuroplasticity. TARKB receptor signaling strengthens. The synaptic plasticity that had been collapsing under chronic metabolic stress and inflammation begins to be restored. The brain's fundamental capacity to learn, to remember, to form new connections, returns. The cognitive restoration that follows is not subtle. It arrives as a return of clarity. The afternoon fog lifts. The word that had been dissolving mid-sentence becomes accessible again. Memory sharpens. Not the deep memory loss of dementia, but the subtle erosion of clarity that had been advancing year by year begins to reverse. Mood stabilizes as the neuroinflammatory state recedes. Executive function clarifies. Attention span lengthens. The sense that your mind was not quite yours anymore gives way to the recognition that your mind is returning. Why this reversal is not theoretical is essential to understand. This is not speculative mechanism. Multiple lines of evidence, from metabolic studies measuring insulin sensitivity, to neuroimaging showing changes in cerebral glucose uptake, to direct cognitive testing before and after metabolic intervention, demonstrate that reversal of insulin resistance correlates with measurable reversal of cognitive decline in adults over 40. The evidence is there. The pathway is established. What remains is the recognition that this is not a disease you live with. This is a metabolic condition you reverse. The fog you have been living in. The word dissolving mid-sentence, the afternoon collapse, the sense that your mind was slipping away, had a name all along. It was not the inevitable march of aging. It was not the slow dimming that you had accepted as the price of your 40s, your 50s, your 60s. It was insulin resistance, settling quietly into your neurons, cutting off their access to fuel, triggering an inflammatory cascade that corroded the very connections your memory depends upon. You now understand the mechanism. You can trace it backward through your own brain. The glucose that circulates in your blood cannot enter your neurons because the transporters that should deliver it have stopped responding to insulin's signal. The energy crisis that follows is not imaginary, it is biochemical. Your neurons, starving in the midst of plenty, activate your brain's immune cells. Inflammation spreads, tau proteins tangle, synaptic connections weaken. The neuroinflammatory state that had been advancing silently for years begins to feel like the end of your cognitive life. But you also understand something else now. This is not irreversible. This is not the destination. This is the threshold. The reversal begins the moment you understand what created the problem. Your brain has not lost its capacity to heal. It has been waiting for the metabolic conditions that make healing possible. Those conditions are not complex. They do not require pharmaceutical intervention or dramatic lifestyle overhaul. They require a return to the way your metabolism actually functions, a way that aligns with how your cells are designed to respond, how your neurons are designed to feed themselves. When you reduce the refined carbohydrates and processed sugars that have been driving constant insulin demand, something shifts. Your insulin levels begin to normalize. Over weeks, the excess circulating insulin that had been hammering against increasingly deaf cellular receptors begins to retreat. Your cells begin to listen again. In your liver and muscle tissue, in your neurons themselves, insulin signaling becomes responsive. The molecular gates that had been locked begin to unlock. GLUT4 transporters relocate to your neuronal membranes. Glucose enters your brain cells again. The paradox that had defined your cognitive fog, blood glucose abundance paired with neuronal glucose deprivation, dissolves. Your neurons are fed. The energy crisis lifts. But the reversal does not stop at glucose uptake. As your neurons are no longer starving, as the chronic energy stress that had been activating your microglia begins to ease, the neuroinflammatory state itself reverses. The sentinel immune cells in your brain quiet. The inflammatory cytokines that had been corroding your synapses decrease. The chronic fire that had been burning through your cognitive reserve begins to cool. And as it cools, the tau pathology reverses. The hyperphosphorylated tau that had accumulated in your neurons begins to dephosphorylate. The tangles that had been strangling your synaptic connections do not disappear instantly. but they stop accumulating. Your brain begins to clear what had been poisoning it. The amyloid beta pathology slows, the vicious cycle between tau and amyloid, where each pathology feeds the other, is interrupted. Your brain's own clearing mechanisms, long suppressed by chronic inflammation, become active again. And BDNF, the molecule that fertilizes neuroplasticity, begins to increase. Your brain's capacity to learn, to form new memories, to rewire itself, returns. The cognitive restoration arrives as clarity. The fog lifts first in subtle ways. You notice you are finishing your sentences. The afternoon does not collapse at 3 o'clock. Words that had been sliding away from you become accessible again. Your memory sharpens. Not the dramatic return of lost memories, but the restoration of the clarity you had lost gradually over years. Mood stabilizes. Your attention span lengthens. The sense that your mind was slipping away gives way to the recognition that your mind is returning. You are not fighting inevitable aging. You are correcting a metabolic miscommunication. The evidence is there. The pathway is established. The mechanism is understood. What remains is the recognition that stands before you now, at this threshold moment. You are not the passive victim of neurodegeneration. You are the agent of your own restoration. Your brain is waiting. The capacity to remember, to learn, to be fully present in your own life. It has not gone anywhere. It has been waiting for you to trace the glucose story backward through your own neurons, to understand what had been happening, and to restore the metabolic conditions that allow healing to begin. That moment is now. The investigation is complete. The path forward is clear.